Welcome once again to my class. I'm going to talk about the diversity of life. Basically, the diversity of life is also known as the biodiversity. And so you can see that we have some images on the screen. We have two types of bacteria, the Archaebacteria and the Eubacteria. That is EU. Then before you spell bacteria, the true bacteria. Then of course we have the protists, the fungus, the plant, and the animal. The both kind of bacteria are found in kingdom Monera. The protist is found in kingdom Protista or Protoctista. Then of course the fungus in kingdom Fungi, the plant in kingdom Plantae, the animals in kingdom Animalia. And this image shows you the different sizes of life forms. Some are very, very microscopic that you cannot even see them with the aid of a compound microscope. You have to use an electron microscope. Then there are some you can see with the aid of your normal compound light microscope. And then with the human eyes, you can see some other types of biodiversity. And so the earth is host to millions of living organisms, millions, different kind of species. They are ranging in different sizes, structures, and functions, from your tiny bacterium to your large whale. They all have lives. If anything can show the 7 signs of life, which you did in secondary school, if you remember Mr. Niger D, then that thing is considered living. But if it shows 6 out of those 7, it's still not alive. Scientists have agreed that for a thing to be considered living, it must show the 7 characteristics of living things, which is of course your movement, respiration, and the others you know about. Then there's a problem of, okay, we have different organisms, but how do we classify them using their forms, their shapes, their similarities, and all of that? And so that is when we come to the concept of taxonomy. Taxonomy is the scientific approach of classifying organisms. So, for example, you find that there are some students in 100 level, some students in 200 level, some students in 300 level, some students in SS1, some students in KG2, for example, using their age and their intellectual prowess or achievements to classify them into different classes. It's the same thing we do with life forms or living organisms, using their sizes, their shapes, their similarities, or their key features or characteristic to classify them into different hierarchy. And so what do we use to classify them? The hierarchy of classification is the kingdom, phylum or division, class, order, family, genus, and species. There's a mnemonic for this. It's called keep ponds clean or frogs get sick. Keep ponds clean or frogs get sick. So kingdom, keep, pond, phylum, clean, class, order or family, frogs, get, genus, and the species, sick. So if you remember that mnemonics, you should be able to remember the hierarchy of classification of living organisms. And of course, this classification system was done or introduced by Carl Linnaeus from he lived from 1707 to 1778. And organisms within a taxon have one or more features in common. The kingdom has the largest variety of organisms. For example, all animals are found in the animal kingdom. All form of plants are found in the plant kingdom. All fungi are found in the kingdom fungi. All bacteria are found in Monera. So the kingdom, therefore, is the largest variety of organisms. It's the highest hierarchy, followed by the phylum, the class, the order, and the others. Then organisms of the same species are closely related. When we say these are organisms of the same species, it means that they can interbreed. When they mate, they can give birth to fertile offsprings that can continue to pass down traits. If they give birth to sterile offspring, then basically they might not be species. They might be of the same genus, but they might not be of the same species. So that's when we're talking about population, for example. We are talking about the total number of organisms of the same species living in a particular area and at a particular time. Okay, so the binomial nomenclature is used to name organisms on the basis of their genus and the species. So instead of saying these organisms from kingdom this, phylum this, the class this, or this, just pick the name of the genus and the name of the species. So we have Panthera leo, for example, for lions, and then we have Homo sapien for humans. And so Homo is the genus, sapiens is the species. So the first name is the name of the genus and the second name is the name of the species. So what you want to do is to always remember that when you are writing the binomial nomenclature for a particular organisms, you have to underline them if you are writing. But if you are typing them, it must be in italics. That means it must be italicized. Then the name of the genus must start with a capital letter, and then the name of the species will all be small letters. Okay, and so these are further examples of living organisms. Piscium sativum, your pea plants, your Rina tigrinia, your Rosa indica, your rose plants, and the rest, the Homo sapiens and the frogs and all of that. And so the common name for an elephant, and then this is a scientific name. You can see the name of the genus and the name of the species. You can see that the genus started with capital letter L, and you can see that the species started with a small letter A. All of these might sound minute or minimal, but I tell you, they are very, very important for you to get your grades, okay? And so we're talking about the five kingdoms. I think I made mention of them earlier when I started. And so we talk about the different kingdoms. If you remember, you have 5 fingers on your palm or on your hands. You should know that we have 5 kingdoms of living organisms, and it was introduced by R.O.H. Whittaker in the year 1969. So please do not confuse the 7 levels of hierarchy, the keep ponds clean, founded by Carl Linnaeus, with the 5 kingdoms principle founded by Whittaker. And so you can get a lot of information from this particular write-up on your screen. The kingdom Monera, of course, the single-celled organisms, unicellular organisms, your bacteria. They can either be motile or non-motile. That means they can move or not be able to move. Normally, if they have a flagellum is the singular, they can move from one place to another. But if they do not have a flagellum, then they are non-motile. In other words, they cannot move. And so some of them have green pigments, and so they can carry out photosynthesis rather. That is your cyanobacteria, your blue-green algae. And so these are different examples there. Then you have to understand that the only kingdom that is a prokaryote or that is prokaryotic is kingdom Monera. It means that they do not have true nucleus or a well-defined nucleus. But the other 4 kingdoms are all eukaryotic. So another way you want to differentiate these kingdoms is in terms of number of cells. Monera is unicellular, Protista is unicellular, fungi is both multicellular and unicellular. Then the last two, the plantae and the animalia, are both multicellular. So in terms of number of cells, we have unicellular as the kingdom Monera and kingdom Protista. Then we have the bridge between unicellular and multicellular as kingdom fungi. Then of course the plantae and the animals must be multicellular. And so, but in terms of a well-defined nucleus, we only have a prokaryotic kingdom, which is the Monera, and the other 4 are all eukaryotic. They have true nucleus. So you can read up on the different characteristics. At least you should know 3 or 4 key characteristics for each of these kingdoms. Viruses, they are like the bridge between living and not living. There are some scientists that claim that viruses are alive, while the other group states that viruses do not show the 7 signs of life, and for this reason, they are not alive. They are basically how do I classify them? Imagine a particular DNA or RNA wearing a protein coat. That is basically what viruses are. They are cellular, and they are biological entities, but they do not have cellular structure or organelles like the endoplasmic reticulum, like the ones we have. Instead, what they do is that they hijack the cells of other living organisms to make their own proteins. They are not capable of making proteins for themselves, although they have RNA and DNA, but they don't have the mechanisms, the other organelles that will be able to kickstart the process. So they have to hijack other cells and force other cells to make proteins for them. And this is why they become dangerous, because they begin to multiply and infiltrate or invade other cells. And before you know, they have, you know, they become infectious. And that is why somebody can be sick. The HIV, the coronavirus, and the Ebola virus, and all of that. So they are just basically packets of DNA or RNA, which are your hereditary materials, wrapped in coats of protein. Okay, so these are all the different types of viruses, and you also need to understand that the virus outside the host cell is a non-living particle called a virion, but when it's inside the host cell, it becomes a virus. So know the difference between a virion and then a virus. Okay, that's it. The general features of a virus: they do not have cells, like I've said earlier. They replicate or multiply, but they do not grow, and they can only replicate or multiply within the cell of a host. And they are obligate intracellular parasites, so you cannot see them without the help of an electron microscope. The same virus was coined by Pasteur, and it's in the Latin word for poison. Like I told you before, they are like thieves, armed robbers. They come in and they force the cells to create proteins for them. This is a size comparison of a virus and bacterial cells. You can see how large the bacterial cells are compared with the viruses. The components of viruses, they either have a RNA or DNA, but never both. They have a protein coat called a capsid. Some viruses have a lipid envelope or membrane surrounding a nucleocapsid core. And the source of that envelope is from the membranes of the host cell. Some viruses package enzymes, while some do not. These are different examples of viruses. One is naked, the other one is enveloped. The life cycle of a basic virus will include that the virus will first of all attach itself to a host cell, then it jets its DNA or RNA, then invade the DNA or RNA of the host cell and forces them to make proteins for them. And that is what they call replication. And the particles of the virus created by the cell comes together to form new viruses, like an assembly of new viruses, and these new assembly attack other cells within the host until they overcome. Thank you for listening and watching my video. I hope to see you next time in our next class.